Okay, good morning. It's always sunny in San Diego. Thanks to the MDS Foundation, Rena Moshi, for the opportunity to speak. I'm going to be talking about genomics, but in this environment, I'm neither conservative or progressive. I'm a moderate when it comes to MDS. These are my disclosures. And I like to do my acknowledgments in the beginning of a talk so I don't rush through them and I don't forget, because without the people on this slide, none of the work is possible. I'd like to especially thank my laboratory, Melissa Fisher, who's my right hand, and Alex Silver, who's a brilliant medical student, MD PhD student working with me. I'll show you a little bit of his data today. We've got a growing group of labs at Vanderbilt. Brent Farrell is a really bright physician scientist doing clonal dynamics, and we've recently brought Alex Bick from the Broad, who's been phenomenal with respect to his contribution to understanding of CHIP. Thank all my funding sources, and of course the patients, the families, and the mice that we inject those patient cells into. Okay, so clonal hematopoiesis is a phenomenon that everyone in this room has at least heard of and familiar with to some degree. It's a phenomenon of aging. It's a hematopoietic stem cells divide, and they do this thousands of times in your lifetime, and there are errors that occur, and sometimes the errors don't get fixed. And if those errors occur and don't get fixed in areas that provide a proliferative advantage, you can grow out a clone. And everyone has low-frequency clones. Everyone in this room has very, very low-frequency clones. The presence of clones where they start to actually provide a potential phenotype and have an impact that they can sustain, we say around 2%, is what we're talking about when we talk about clonal hematopoiesis of indeterminate potential, or CHIP. And these mutations occur in the same genes that we see in MDS, epigenetic function, splicing function, DNA damage repair, and we'll talk about them briefly. But this is in a very important discovery. It's been a decade since this discovery was made, and we have a good sense of what the molecular epidemiology is of clonal hematopoiesis right now, where 3 to 6% of 60-year-olds and probably 10% of 70-year-olds have CHIP. And the CHIP genes are the same genes, as I said, that we see in MDS. And these CHIP mutations increase risks of lots of disease, cardiovascular diseases of all kinds, peripheral vascular disease, stroke, renal disease, pulmonary disease. We're going to concentrate more on myeloid transformation in this talk, of course. But the molecular epidemiology of the mutations that we see in CHIP is different than what we see in MDS. DNMT3A is about half of all the CHIP mutations, but DNMT3A is the 6th or 5th most common genetic aberration in MDS. Likewise, SF3B1 accounts for only about 3% of CHIP, but it is the most common aberration we see in MDS. Therefore, some mutations are a little bit potent, if I can say that word, a little more potent than others in leading to a risk of developing myelodysplastic syndrome. We have some ideas of why this is. We study these gene mutations and the effect they have on the immune cells. We know that DNMT3A mutant monocytes increase the constituency and the amount of cytokines, inflammatory cytokines that are secreted. It changes the interaction between T cells and monocytes, and a change in interaction between the vascular endothelial monocytes. It's not just monocytes. There's increased expression of integrins on neutrophils that slow down leukocyte adhesion and rolling, and lots of other contributions from other cells that are made in the bone marrow, but monocytes seem to be where a lot of the activity is. Likewise, in our lab groups, this is Brett Heimlich and Pavan Bhatt, who looked at patient samples from our repository of patients with CHIP and found that the TET2, the monocytes with TET2 VAF that was high, a considerable VAF, were more prone to inflammatory signaling as measured by phosphostats 3 in both a non-stimulated and a stimulated assay. So we have this concept of age association with CHIP and a progression to a phenotypic pre-disease state like clonal cytopenias of undetermined significance, CCUS, clonal monocytosis of undetermined significance, CMUS, or this is a tough one, cytopenias in the presence of clonal monocytosis of undetermined significance, or CCUS. And these states, we have the notion that they increase the risk of developing MDS and ultimately AML. Well, Luca Moccovati's group was the first to really kind of go after, the specific risk formally of developing myeloid neoplasms from a clonal cytopenias of undetermined significance. And in their cohort, this is a refined analysis published 4 years after the original paper, but this is a very informative paper published a couple of years ago where Luca's group illustrated that in an unsupervised cluster analysis, they found that patients who had singular DTA mutations were at very low risk, actually, of developing myeloid neoplasms. But if patients had other more, to quote Charlie Schiffer, MDS type mutations, or they had combinations of mutations or high VAFs, they were more likely to develop myeloid neoplasms. And now we're looking at this as probably a variant specific, which is terrifying. This is Alex Silver, who I briefly mentioned in my acknowledgement slide in the beginning. And Alex is a new father, so he can't be at ASH to present this data himself. But he discovered that the R882H mutant has a completely different phenotype from a cardiovascular risk perspective, both in big data sets and in molecular models than the R82C, the one residue change. So think about that. A lot of our scoring systems, we're talking about binary. Yes, you have the mutation. No, you don't have the mutation. We're graduating now to saying VAF matters, but it actually matters specifically at which residue. So that's a big multiplier on how we think about genomic risk in clonal hematopoiesis and transformation to MDS. This year, Lachelle Weeks and Ben Ebert's group in Boston published this really elegant prognostication tool in the New England Journal of Medicine Evidence. And I encourage you to go to chrsapp.com and check this out because it's really helpful. We use it in our CHIP clinic. The first thing they did was basically edify our early conclusions that CCUS does lead to an earlier, a greater chance of developing myeloid neoplasm than CHIP. This is looking at, UK Biobank, which is a population database in the UK with over 450,000 participants. And these patients have been followed for 10 years, which is particularly helpful when you're studying something like CHIP, which takes time to develop into disease. In their analysis, the same kind of factors we were thinking that caused increased risk of transformation in leukemia or progression into MDS and CMML, neoplastic states. Age, the type of mutation that we see, whether or not they had CCUS or just CHIP. The VAF, was the VAF greater than 20%? The number of mutations. But they also found some specific blood markers that we've known for years about, but didn't know they had such an impact on determining the risk of developing MDS from CHIP. This is the RDW, the red cell distribution width, and the MCV, the mean corpuscular volume. So they did this, a Cox regression off this, what's called recursive partitioning based on the incidence of myeloneoplasm. So they took all of these patients and in this database, and there was about 207 of them that developed, I think it was 207, over 200 patients who developed myeloid neoplasm, and then partition them on high-risk mutation, no high-risk mutation. In the green illustrates a lower risk to develop MDS, and the red is a higher risk. And what you could see is pretty quickly, if you have a high-risk mutation and a high RDW, you get a 50% chance of developing MDS in the presence of CHIP, which is remarkable with respect to how quickly you can make that determination. If you follow that down, patients who have a high MCV and have a high RDW and have a high-risk mutation, it's a foregone conclusion that they're likely going to develop MDS. On the other hand, if those patients with high RDW then have a low MCV and only one mutation, their risk is limited. And we use this tool in our CHIP clinic when we're talking to patients and thinking about how to follow them. So this model partitions very well, and I encourage you to go to, I won't spend more time talking about the risk model, but it is an important advance. And if you go to this website, chrsApp.com, you can play around with the tool, much like the MDS-risk-model.com for IPSSM. Everything is now a .com available, so that's great. So I'll spend the latter part of this talk talking about our biorepository and registry for clonal hematopoiesis, which we call CHIVE, because everything at Vanderbilt's got to have a V in the acronym, and that's the best we could do. So clonal hematopoiesis and inflammation in the vasculature is a registry and biorepository, a collaboration with a vascular specialist thinking about inflammation and clonal hematopoiesis. The registry is for patients with clonal hematopoiesis, and here's something that's a little different than what you might think. We actually can send patients who are at risk for CH. And we define that as anyone over 40. So if you're over 40, you're at risk. Anyone over 18 with any disease that is found to be increased risk in the presence of CHIP, like coronary vascular disease. So as you'd imagine, outside of the pediatric hospital, this is most of the people who come through the door. So we're working on trying to screen in various different clinics around the hospital. We have a database that's EPIC informed, so it's not a complete carbon copy of the medical record, but it has a lot of discrete data that's captured. The biorepository is sequential, so patients come in at a set period of time and their blood samples are drawn, and if they have a bone marrow biopsy, then the bone marrow is recovered. And of course, there's IRB approval to do a variety of different genotyping and cellular assays in those samples. We developed something called CHIVE-seq, which is a Twist Bioscience hybrid custom capture assay. And we reasoned that one of the main stumbling blocks or rate-limiting steps for doing a large-scale genotyping experiment was the cost. So we chose to use, instead of a whole exome, whole genome, a greater depth at where the juice was worth the squeeze, so to speak. So 95% of all reported CH mutations are covered within this panel. And we do this at about 1/10 of the cost of traditional commercial sequencing panels. So I'll show you the first version of our first analysis we've done, which is about 200 patients. The patients were enrolled either from CHIP clinic, we have a CHIP clinic, and we'll talk a little about that, or they're enrolled due to being at risk for CHIP, from the cath lab, from renal clinic, or what have you. And then they're sequenced with CHIVE-seq, and they're put into two groups, the CH group and the non-CH group. And what's, I won't spend a lot of time talking about it because of the limited time we have, but what's interesting is patients come into CHIP clinic, and those of you who run a CHIP clinic, you know how this looks. Patients come into your CHIP clinic referred with quote-unquote CHIP, and oftentimes they have a polymorphism, a germline pathogenic mutation, or some mutation that just hasn't been called properly, or just isn't CHIP. And it's created kind of a little bit of a Pandora's box with some of those patients, but it's a good learning experience when doing this kind of analysis. So if you look at the CHIP and no CHIP, as you'd imagine, there's more male, they're older, and although we don't have the numbers to show it, we think there's a trend towards a higher BMI in those patients, which makes sense with respect at least to the vascular risk. The molecular epidemiology that we see is consistent with what's been done retrospectively. This upset plot just illustrates that about half the patients have singular DNMT3A or TET2 mutations, but a full 55% of the patients have DNMTA single or double mutations. When you look at objective criteria, clinical features between CH plus and CH minus patients, you can see that the increased risk of chronic kidney disease, but really not yet at this juncture a difference in our blood counts or inflammatory markers to speak of. And then when we looked at cardiovascular endpoints, we saw an increased risk of hypertension, heart failure, and coronary artery disease consistent with what we know about CHIP, but not some of the objective markers like BNP and ejection fraction and blood pressure that make that up. What's probably interesting to this group is we found 6 patients in just a 1.5 year follow-up time that progressed from clonal hematopoiesis to malignancy. And just like the CRHRS would tell us, Five of these patients either had multiple mutations, high-risk mutations, or mutations with high allele burden. And there's one patient here with the TET2 patient at the bottom with 2% allele burden. And we're trying to investigate, okay, well, why did this person develop myeloid neoplasm? Because according to CRHS, this person has a less than 5% chance over 10 years of developing myeloid neoplasm. So this prospective tool allows us to validate what we know, but also give us an opportunity to discover some new influence from perhaps the germline that may lead to that mutation. We follow a rubric that's informed by CHRS. Because CRHS, we're probably going to modify our rubric a little bit, but basically if we think someone has a high-risk mutation, we follow them every 6 months. If we think someone has a lower-risk mutation, we follow them a year. If they don't have CH, but they're in the protocol, we continue to draw blood once a year because we want to capture those patients who are at risk for CHIP from their pre-CHIP to their CHIP sample to see if we can start to discover some of the differences that occur molecularly and the changes that occur molecularly to lead to that emergence of that clone. So in summary, clonal hematopoiesis is, make no mistake, this is one of the biggest discoveries in this millennium, and I think that it's a great opportunity for us to understand biology and make some big impact for new therapies. We have an early prospective analysis that seems to cooperate with what's been done retrospectively. But this really has to be done at multiple centers. It has to be done in great volume. It has to be done multicultural and different lineage and backgrounds to really truly understand risk. So I'll end with a plug for the next annual meeting of somatic mutations and pre-disease. We'll talk about addressing high-risk clones in Nashville in the fall of 2024.
Wonderful talk. Thank you very much. Are there any questions?
Can I start?
Sure.
Michael, I have a question. I've been asked by the regulatory authorities once in a while, why to identify these patients, with pre-MDS states? Do you have anything to do with it? Once you diagnose, so what? Can you offer a treatment or therapeutic approach that will delay the development of the disease?
Well, yeah. I mean, this is the recursive partitioning has a purpose because we have to learn who are the patients who are going to actually get disease, who are the patients who are at high risk with clonal hematopoiesis. And there's already been a variety. If you go to clinicaltrials.gov and search on clonal hematopoiesis or CCUS, you'll see there's a variety of interventions. But as you would imagine, the regulatory authorities in the United States, just like in Israel, are not going to want you to treat someone with a potentially toxic chemical who doesn't have a disease. So these are vitamin C. In some cases, they're higher risk CHIP with an inhibitor of a certain mutation, like an IDH mutation. But for sure, both in myeloid disease and vascular disease, there's promise for secondary and tertiary prevention. The CANTOS study looked at the use of canakinumab in cardiovascular disease. Across the board, canakinumab had a 10% drop in all-cause cardiovascular outcome. I think that's the number. But if you look at TET2 mutants in that study, in a post-hoc analysis, the reduction in cardiovascular and at the cardiovascular endpoint was 70% with the use of an IL-1 receptor beta antagonist. So there are opportunities, but it's not quite the same equipoise that we're used to as MDS doctors. We're not talking about a neoplasm, which is going to kill you in a matter of a year to 10 years, depending on what kind of MDS you have. We're talking about a patient who may develop a neoplasm. So it's upon us to try to understand which of these patients, their germline background, their inflammatory milieu, their blood counts, their RDW, MCV, whatever it is, who are going to be at the high risk, and how do we develop therapies for them? And I will note that as you'll learn at this meeting, a lot of the new development in MDS is around niche inflammation. So this is a kind of a groundswell that we're starting to kind of see, targeting, IRAC, signaling and so forth to slow down this feed-forward progress of inflammation that we see in early low-grade MDS and comes in the setting of CCUS.
Michael, I'm going to ask you another question. When you're looking at cardiovascular event rates, are you adjusting for the use of disease modifiers like statins, in the use of statins in these patients that may reduce their risk?
Our prospective analysis? Yeah, I mean, this is a pretty simple analysis. We just wanted to get this out there and say, hey, we're doing this. Let's get people on board. I guess my time's up. We'll get people on board at many other centers. When the number grows, we can start really controlling for more of these things and make sure we're looking at the apples and apples and oranges and oranges.
Thank you.
Thank you.
Thank you, Michael.